Good day everyone, good day viewers. I am Mary Jane T. Katig and I am going to tackle the three parasites, which is the Diphyllobothrium latum, Echinococcus granulusus, and the Entamoeba hystolytica. So now, we are going to tackle what is their life cycle, epidemiology, distribution and transmission, diagnosis, symptoms, treatment, and prevention. So first is the Diphyllobothrium latum. Diphyllobothriasis represents an intestinal parasitic zoonotic infection caused by the cestode Diphyllobothrium. Diphyllobothrium latum, which is the most common cause of Diphyllobothriasis, also called the fish tapeworm or the broad tapeworm, is transmitted to humans by the ingestion of fish which harbor infectious larvae of the genus Diphyllobothrium, causing a wide-ranging spectrum of disease and severity. Diphyllobothrium latum belongs to the kingdom of Animalia, phylum Platyhelminthes, class Cystoda, order Pseudophilidia, family Diphyllobothridae, genus Diphyllobothrium, species Diphyllobothrium latum. So next is the life cycle. Eggs are passed and embryonated in feces. Under appropriate condition, the eggs mature with approximately 18 to 20 days. In yield oncosphere, which develop into a poricidia after ingestion by a suitable crustacean, or the first intermediate host. The poricidia develop into porcicoid larvae. In the next stage, porcicoid larvae are released for the crustacean upon predation by the second intermediate host, or which is usually a small fish, and migrate into the deeper tissues where they develop into a plerocercoid larvae or spargana, which is the infectious stage for the definitive host. Next stage, because of humans do not generally eat this small fish species raw, the second intermediate host probably does not represent an important source of human infection. However, this small second intermediate host can be eaten by larger predator species than then serve as paratenic host. In this case, the plerocercoid migrates to the musculature of the larger predator fish, humans, to acquire the parasite via consumption of undercooked paratenic host fish. In the definitive host, the plerocercoid develops into adult recruits in the small intestine. Adult Diphyllobothrium attached to the intestinal mucosa by means of two bilateral grooves, which is guatria, of their stomachs. The adults can reach more than 10 meters in length with more than 3,000 prognaths. Immature eggs are discharged from the prognaths, from up to 1 million eggs per day per worm, and are passed in the feces. Eggs appear in the feces 5 to 6 weeks after infection. Next is the epidemiology, distribution, and transmission. The person who regularly consume raw fish are affected by the Diphyllobothrium latum. Diphyllobothriasis can affect any age group in general, but the majority of identified cases were middle-aged men. This parasitic infection can be a public health issue for residents and travelers in endemic as well as non-endemic regions. The fish tapeworm, Diphyllobothrium latum, has historically been implicated in human illness for thousands of years. The current state of knowledge regarding this parasite points to cohabitation with humans since the early Neolithic period. Epidemiological studies have concluded that in the 1970s, this infection was estimated to have affected 9 million individuals globally, with newer data estimating 20 million people currently infected worldwide. Additionally, in recent years, certain areas of the world have seen a re-emergence of Diphyllobothriasis, thought to be secondary to changes in eating habits as well as globalization. Furthermore, it is postulated that with changes in eating habits combined with rapid freezing techniques, there will be an increased rate at which potential fish-borne tapeworm infections will start to arise. Since Diphyllobothriasis is mostly a function of consumption and contact with fish, specific individuals are at higher risk of disease transmission. This includes fishermen who consume their fresh catches and individuals who consume and cook fish regularly. Next, Diphyllobothrium latum causes the reduced host intake of vitamin B1, resulting in megaloblastic anemia. Like other tapeworms, it can cause local inflammation in the intestine as a result of physical presence. It is made by identification of eggs or segments of the tapeworm in a stool sample with a microscope. Eggs are usually numerous, but more than one stool sample may be needed to find them. Complications include intestinal obstruction and gallbladder disease caused by the migration of microbes. Next, most infections are asymptomatic. However, symptoms can include abdominal discomfort, diarrhea, vomiting, and weight loss. Vitamin B12 deficiency leading to pernicious anemia may occur. Prevention: thoroughly cook all fish or don't eat fish. The treatment for these parasitic infections, as well as most tapeworm infections, is praziquantel in those without contraindications. A 25 mg or kilogram dose is noted to be highly effective against Diphyllobothrium latum infections. So next, these are the references. So let's go on with Echinococcus granulusus. Called as the hydatid worm. Human echinococcus is caused by the larval stage of cestodes, or the tapeworms, of the genus Echinococcus granulosus. It causes cystic echinococcus and is the form most frequently encountered. Another species are Echinococcus multilocularis, or causes alveolar echinococcus and is becoming increasingly more common with Echinococcus granulusus. It has three parts, which is the scolex, pyriform 300 micrometer, 4 suckers, and a putrucible rostellium with 2 circular rows of cucullins. Next is the neck, short and thick. And lastly, the estrobilia, with 3 to 4 proglottids, usually immature and graphic. It is belong to the kingdom of Metazoa, phylum Platyhelminthes, class Cystoda, order Cyclopelludia, family Echinidae, genus, which is the Echinococcus, and the species Echinococcus granulosus. The adult Echinococcus granulosus is measured as 2 to 7 mm long. From the first stage, it resides in the small intestine of the definitive host. Gravid proglottids release eggs, then that are passed in the small intermediate host, eggs hatch in the small intestine and release seeds with oncospores that penetrate the intestinal wall and migrate through the circulatory system into various organs, especially the liver and the lungs. In these organs, the oncosphere develops into a thick walled hydatid cyst that enlarges gradually. Parasite is transmitted to dogs when they ingest the organs of other animals that contain hydatid cysts. The cyst develops into adult tapeworms in the dog. Infected dogs shed tapeworm eggs in their feces, which contaminate the ground. The most common mode of transmission to humans is by the accidental consumption of soil, water, or food that has been contaminated by the fecal matter of an infected dog. Echinococcus eggs that have been deposited in soil can stay viable for up to a year. The disease is most commonly found in people involved in raising sheep as a result of the sheep's stool as an intermediate cause of the parasite and the presence of working dogs that are allowed to eat the offal of the infected sheep. Sheep, cattle, goats, and pigs ingest tapeworm eggs in the contaminated ground. Once ingested, the eggs hatch and develop into cysts in the internal organs, which becomes the transmission of Echinococcus granulosus. Next, Echinococcus granulosus oncosphere develops into cysts, which grow slowly, usually over many years, into large unilocular fluid-filled lesions, hydatid cysts. Root capsules containing numerous small infective protoscolices form within these cysts. Imaging techniques such as CT scan, ultrasonography, and MRIs are used to detect cysts. After a cyst has been detected, serologic tests may be used to confirm the diagnosis. Next, Echinococcus granulosus infections often remain asymptomatic for years before the cysts grow large enough to cause symptoms in the affected organs. The rate at which symptoms appear typically depends on the location of the cyst. Hepatic and pulmonary signs are the most common clinical manifestation, as these are the most common sites for cysts to develop in addition to the liver and lungs. Pain in the upper right part of the abdomen, the liver cyst, increase in the size of the abdomen due to swelling, bloody spotting, chest pain, cough, and severe allergic reaction or anaphylaxis when cyst breaks open. How can it be prevented? It can be prevented through washing your hands with soap and warm water after handling dogs and before handling food. It can be treated or managed in the option range from expectant therapy to invasive surgical intervention. The choice depends on the variety of factors, including the patient's clinical picture and the size of the cyst, which is going to be seen in the ultrasound-guided staging system, which is the method used in the disease. Next slide, these are the references. So, let's move on to Entamoeba hystolytica. Parasite is a common protozoan parasite found in the large intestine of humans. The parasite is responsible for the amoebiasis and liver abscesses. It is the third leading parasite cause of death in the developing countries. It has three types: the trophocyte, the precyst, and the cyst. The trophocytes are from 20 to 30 micrometer in diameter and contain a vesicular nucleus with a central endosome, peripheral chromatin, and radial achromatic fibrils, imparting a cartwheel appearance. So, it belongs to the kingdom of Protista, phylum Sarcomastigophora, class Lobosea, order Amoebida, family Sarcodina, genus Entamoeba, and species Entamoeba hystolytica. So, trophocyte, it is the growing and feeding stage of parasite, shaped not fixed because because of constantly changing position. The pre-cyst, it is the intermediate stage between trophocyte and cyst. It is the smaller in size from 10 to 20 micrometer. Meanwhile, the cyst is the infected form of parasite. Shape, it is round or round or oval in shape. This non-pathogenic Entamoeba dispar, however, is morphologically identical to Entamoeba histolytica, and differentiation must be based on isoenzymatic or immunologic analysis. Molecular methods are also useful in distinguishing between Entamoeba histolytica and Entamoeba dispar. The life cycle of Entamoeba histolytica is relatively simple and consists of infective cyst and invasive trophocyte stage. Life cycle completes in single host. From human, when it gets infected by Entamoeba histolytica cyst from contaminated food and water. Infection can also acquire directly by anogenital or orogenital sexual contact. The mature cyst is resistant to low pH of stomach, so remain unaffected by the gastric juices. The cyst wall is then lined by intestinal trypsin, and when the cyst reaches the caecum or lower part of ileum, excistation occurs. The neutral or alkaline environment as well as the bile components favor excistation. Excistation of a cyst gives 8 trophocytes. Trophocytes are actively and carried to large intestine by peristalsis of small intestine. Trophocytes then gain maturity and divide by binary fission. The trophocytes adhere to mucous lining of intestine by lectin and secrete proteolytic enzymes, which causes tissue destruction and necrosis. Parasites, when gain access to blood, migrates and causes extraintestinal disease. When the load of trophocytes increases, some of the trophocytes stop multiplying and revert to cysts formed by the process of encystation. These are released in feces, completing the life cycle. In a small number of instances, it has been shown to spread to other parts of the body, such as the lungs or brain, but is a very uncommon symptom of amoebiasis. From about 80 to 90% of infections are asymptomatic and are likely due to the non-pathogenic species Entamoeba dispar or Entamoeba moschkowski. Therefore, the worldwide incidence of Entamoeba histolytica is more likely estimated to 5 million cases annually, with global mortality still at 100,000 persons per annum. The majority of amoebiasis cases occur in developing countries and industrialized countries. Risk groups include men who have sex with men travelers, recent immigrants, immunocompromised persons, and institutionalized population. It is most common in the world in tropics and subtropics in the temperate zone. The habitat is the trophocytes of Entamoeba histolytica that lives in the mucus and submucous layers of the body. The mode of transmission is from fecal-oral route, ingestion of food or water contaminated. The parasites express large number of virulence factors, including lectin, lytic peptide, cysteine, proteinases, and phospholipids. Excistation of cysts and intestine releases 4 trophocytes, which then colonize the large intestine. The binding of trophocytes to the colonic epithelium is a dynamic process in the pathogenesis. Trophocytes penetrate the columnar epithelium of mucosa, causing lysis and multistake inside till they reach submucosal layer and multiply rapidly. The diagnosis of amoebiasis can be very difficult. One problem is that other parasites and cells can look very similar to Entamoeba histolytica when seen under a microscope. Therefore, sometimes people are told that they are infected with Entamoeba histolytica even though that they are not. Entamoeba histolytica and another amoeba, Entamoeba dispar, which is about 10 times more common, it looks the same when seen under a microscope. Unlike infection with Entamoeba histolytica, which sometimes makes people sick, infection with Entamoeba dispar does not make people sick and therefore does not need to be treated. Microscopic identification of cysts and trophozoites in the stool is the most common method for diagnosing Entamoeba histolytica. This can be the steam preparation, for example, or the trichome. Concentrate from fresh stool, wet mounts, with or without iodine stain, and permanently stained preparations. Concentration procedures, however, are not useful for demonstrating trophozoites. In addition, Entamoeba histolytica trophozoites can also be identified in aspirates or biopsy samples obtained during colonoscopy or surgery. The majority of infections restricted to the lumen of the intestine are asymptomatic. Amoebic colitis or invasive intestinal amoebiasis occurs when the mucosa is inflated. Symptoms are often quite mild and can include loose feces or poop, stomach pain, and stomach cramping. Amoebic dysentery is a severe form of amoebiasis associated with stomach pain, bloody stools, and fever. Rarely, Entamoeba histolytica invades the liver and forms an abscess, a collection of pus. In a small number of instances, it has been shown to spread to other parts of the body, such as the lung or the brain, but this is very uncommon. Next, eradicating fecal contamination of food and water through improved sanitation, hygiene, and water treatment in non-endemic areas. Disease transmission can be reduced by early treatment of carriers. The following suggested treatment are the diloxanide, chlorate, didohydroxine, paramycin, metronidazole, imidazole, 4-aminoquinoline for extraintestinal infection and oral rehydration. So let us be aware of the food we eat and maintain a healthy and safe places. Generally, Diphyllobothrium latum is an intestinal parasite of humans and other fish-eating mammals and birds like penguins and deer. While Echinococcus granulusus is also called hydatid worm, a cyclophilic cestode that dwells in the small intestine of canids as an adult. While Entamoeba histolytica is an amoeba parasite that feeds on cells in the human colon, it is the cause of amoebic dysentery as well as colon ulcerations. These parasites can be prevented through cooking your food properly and avoiding phrases as what I said. So that's all for today, guys. I hope you have learned and thank you for watching.